Okay, welcome. Everyone hear me okay? Well, thank you for coming. My name is Carl Peters. I'm an application scientist with BMG LabTech. And I'm happy to be here to provide a little bit of introduction for our featured speaker, Daniela. And she'll be talking about some screening applications that she has done in her work at Sanford Burnham. And I just want to take some time to talk about the technologies that went into making sure that the work that she was able to do is successful. And that is going to feature the use of the Ferrostar FSX. So first, a little bit about BMG LabTech, if you don't already know. We are a company that focuses completely on producing microplate readers, so we're very focused in the type of products that we're developing. And we have a lot of experience. The products are all made in Germany and engineered there. We have everything from single mode microplate readers up to multifunctional microplate readers that can perform essentially any type of detection technology that you can think of. So that means that people are using these in basic research labs all the way up through high throughput screening facilities. And since we are focused on microplate readers, we make sure that we have put the technology into that to ensure that we have specifications that are as good or better than any other microplate reader that you can find. So as I mentioned, the home office is in Germany, but there are daughter locations throughout the world, and of course I represent the U.S. office, and with this network and with distributors, we're able to provide worldwide sales and support. So no matter where in the world you are, you'll be able to get access to BMG LabTech readers and the necessary support that you need to make sure that you are getting what you want done with the reader. So as I mentioned, I'm going to be talking about one of our readers in particular, the Ferrostar FSX. This is our top-of-the-line reader, mostly for high-throughput screening type of environments, but can be used for assay development as well. So some of the main features that really make this reader stand out are the fact that it has simultaneous dual emission detection in all the read modes where that would be important. So for fret, Brett, time resolve fret, multiplexing of alpha screen, fluorescence polarization, those all really benefit from having this ability to read the plate once and get both signals at the same time. It is capable of reading microplate formats anywhere from 6 up to 300, 3, 4, 5, 6 well plates. And it has standard incubation and shaking for more kinetic types of assays that you might want to perform. So the kind of key component in a lot of ways that makes the Ferrostar not only very sensitive but very easy to use is the optical setup, and that's displayed here. So the optics are all contained in what we call optic modules, and the idea being that these are assay specific and will allow you to have everything that you need already there. They're bar-coded so that when you put them into the reader, it'll automatically scan them and it will know exactly what capabilities that you have available at any time. Because of the free air optics system that we have, the same module can be used for both top and bottom reading, and so the switch only requires that you to make a change in the software. So one of the assays that Danielle is going to talk about involves luminescence, and as well as some time-resolved fluorescence, and for both of those, optimal performance can be achieved if you are able to focus, to reach a focus point in the well that has the highest signal. And so we have this very sensitive Z-prime adjustment with 0.1 millimeter resolution. So and you can see with this bell curve that reading at the peak is really going to be quite effective. Reading slightly off peak can start to decrease the signal that is possible that you can see. So this is something that can be done from either the bottom or the top to ensure that you're gathering the best signal possible. We do have some automation features. The stacker is for people that are looking at doing sort of mid-throughput types of assays. Within the microplate reader, there are barcodes. So that allows you to communicate the information about what's present on the plate to the software and have that all tracked internally. Further down the line is if you are going through into a truly high throughput situation, we have robotic integrations with essentially every robot company out there. So it's really up to you. It's your choice which type of system you want to work with. We'll make sure that that can happen for you. So some of the things that we have thought about in terms of some of the different types of assays are things like crosstalk. So especially in luminescence and alpha screen types of assays, there's the possibility that a signal from a neighboring well will affect the well that you're trying to read at any given time. And this is primarily an effect that is seen based on the type of assay plate that you're actually using. But if you want to be confident that what you're reading is the true signal that is being produced by that individual well. And so we've taken some measures to alleviate the problems that are caused from crosstalk. And so we've put in these crosstalk reduction spoons, and these will block essentially the light that's coming from the tops of the wells, and allow for the signal that is going to reach the detector to be solely from the well that is being read at that time. As I said, the plate type can often influence the amount of crosstalk. So not only can you see crosstalk through the top of the wells, which we can handle with the aperture spoon, but even between the wells, there can be light coming through depending on the type of plate. So we do have a mathematical approach that can correct for that if that's the type of plate that you're using. The other type of assay that Daniela is going to talk about is some TR-FRET types of assays. We've long been a leader in the detection of TR-FRET and have always had matched PMTs that are dedicated for detecting time-resolved fluorescence. And this is something that has definitely set us apart from other companies in the past. You can use a laser or a flash lamp for the excitation of the time-resolved fluorophore. And we've made some improvements to the laser technology so that it's even faster and able to generate high-quality data in very short read times. And then I want to focus on a little bit this feature called decay curve monitoring. It's not something new. It's something that really has been a part of the Ferrostar for a very long time, but it's something that I think is underappreciated. And so if you're familiar with time-resolved fluorescence, as I'm sure you are, you know that the reason why it's so useful is because the signal is going to be longer lasting. And so you can choose a time to start collecting data that is after where the high background signal is going to be present. And what the decay curve monitoring tool allows you to do is actually to visualize what those curves look like, and then to make your own choices as to what is going to be the best possible selections as far as the interval time and the read start time for your particular assay. And to help you in making those determinations, we have provided in our data analysis software a tool that will actually allow you to select what's most important for you in terms of what parameter of assay quality you want to look at, and then allow you to find within a grid of results what is going to be the best result for you. So in summary, BMG continues to work and develop new things, new features that provide the best sensitivity, speed, and performance. We have a very large dynamic range in luminescence, and we're continuing to improve the sensitivity in all read modes. And I talked a little bit about the existing and some new tools that are going to help you further optimize your assays. So with that, I will pass it along to Daniela.
Okay, so thank you for this opportunity to present some of the research that we did on the development of functional binding and multiplex assays targeting the type 1 cholecystokinin receptor to screen for novel potential obesity treatments. Okay, so this work I have been working on during my time at Stanford Burnham Prebys. I recently moved on from there, but the CCK1R project was a part of a collaboration with the Mayo Clinic. And what I will be talking about is also published in SLAS Discovery in 2022. That's the screening part. And we recently also published a follow-up paper about the mechanism of action of the hits that we found in membranes. So first, I would want to dive a little bit deeper into why we think CCK1 receptor would be a good therapeutic target to treat obesity. Then I would move into the SLAS discovery paper where we talk about the high throughput screening. So we developed a testing funnel and developed assays like the CCK1 receptor IP1 PAM primer screening assay, a binding assay for target engagement using TF-RET, and a sort of multiplex assay that can detect CMP and beta-arrestin in the same well. And for all those assays, we used the Ferrostar FS or FSX reader and some of the modules that Carl has introduced you to. Also part of this paper was the hit validation of original hits in the assays that we had developed. And then I want to spend the last couple of minutes into the scaffold validation of our CCK1 receptor positive allosteric modulators, the mechanism of action studies, and some analog by catalog and structure activity relationships. So with that, I want to start with the background. So cholecystokinin, it regulates appetite. It is a gastrointestinal hormone and is synthesized and secreted from neuroendocrine eye cells after a meal. So when you eat nutrients, especially fats and proteins, it senses it and then it secretes CCK. This then leads to gallbladder contraction, secretion of pancreatic juices, and also regulates the intestinal transit and the gastric emptying. So it just wants to make sure that you get the optimal absorption of the food, and it also induces satiety. This action of CCK is mediated by the activation of the type 1 cholecystokinin receptor, so CCK1R, or also known as CCK-AR, which is present in the gallbladder, pancreas, pylorus, and important for our indication on the periphery of vagal afferent neurons, where it then communicates with the brain and stimulates the satiety and satiation. I just want to point out that there's another type of CCK receptor, the CCK2R or BR. B stands for brain because it's more broadly expressed in the brain. There's a little bit overlap in the intestine, but in general it has very distinct expression patterns to CCK1R. There are multiple forms of CCK, so it's not just one form that's floating around in the human body, there are multiple forms. And it's produced from a single gene by post-translational processing of preprocholecystokinin. The pharmacophore that binds to CCK1R is mainly a 9 amino acid peptide from the C terminus of the whole fragment. Interestingly, the most abundant form in the intestine is actually CCK58, whereas CCK8, the smallest peptide that is recognized and able to fully activate CCK1R, is more broadly found in the brain. And this is important because studies have shown that actually there's a difference in the way how these CCK peptides affect appetite and meal size intervals. So for CCK58, it elicits both satiety and reduces the meal size, and it importantly also prolongs the intermeal intervals, while CCK8 elicits only satiation and reduces this intermeal interval. So that actually would be contradictory for what we are looking for in our indication. I want to point out though, I tried, but CCK58 is very hard to come by, and all the in vitro studies that you're going to find, pretty much all of them are going to be done with CCK8 or a shorter form of the peptide. So I just wanted to point that out, that actually the form that might be more interesting for this indication is the CCK58. So the CCK1 receptor activation is a therapeutic strategy to treat obesity. It's not a new thing. So there have several CCK1 receptor agonists been developed. There have been peptides, peptoids, and small molecules, like the ones from GSK and Merck that you can see on the right. They found compounds with sub-nanomolar potencies and affinity, and they were able to reduce meal size and cause weight loss in animal models and studies, but they didn't progress any further. And this is probably true because they all ran into a challenge, because these highly potent and often also long-acting full agonists, they have shown a lot of side effects. So very common side effects like nausea, abdominal cramping, and diarrhea, but there's also a theoretical possibility of trophic effects, and it could potentially progress and maybe develop pancreatic cancer. So we've been thinking we need a novel strategy for targeting the CCK1 receptor. Our collaborator, Larry Miller, has been doing research on this topic for quite some time, and he's also been thinking about what would be potential ways to develop safer and more effective drugs. So first, I want to go a little bit deeper. How does the CCK1 receptor actually signal? So it's a class A G protein coupled receptor. It's like a peptide GPCR. It has the seven transmembrane domain in the membrane, and CCK or other peptides are the orthosteric ligand, and when it binds, it couples to the different G proteins, GQ, GS, or can also recruit beta-arrestin. In general, it prefers GQ11 proteins, but it can also couple to GS or G13, or recruit beta-arrestin at higher concentrations of CCK. I want to point out that all this has been identified with CCK8, and for the other CCK peptides, like the different forms, the signaling profiles are more poorly understood. That's also why our collaborator thought that looking for biased agonists, what is also a novel way potentially to target CCK1 receptor might not be, we don't have any basis for that at this point because the signaling events are poorly understood, and we don't really know which pathway we would have to look at to actually induce the satiety and satiation that could help us to treat obesity. So we were looking into allosteric modulators. So allosteric modulators bind to structurally distinct binding sites and then can potentiate the effects of endogenous ligands like CCK8, but also, for example, CCK58. They also are characterized by probe dependency and saturability of effect, which I will show in a later slide. Interestingly, in this cartoon, we also see here in yellow, which is marked as lateral allosteric modulator, we see cholesterol. And this is because our collaborator, Larry Miller at the Mayo Clinic, he found that the CCK1 receptor signaling is impacted by elevated cholesterol levels. So here on the left side, we see studies on CCK1 receptors that are expressed on a regular CHO cell or on cell lines that have been shown to have increased membrane cholesterol levels. And they found that there's a reduced response to CCK, so they are shifted to the right in the cells with higher cholesterol levels. This is very specific for CCK1R. We didn't see this effect with the type 2 cholecystectonin receptor. Our collaborators also studied this phenomenon further and found that the tyrosine 140 is a key residue for cholesterol sensitivity. And they even found that when you mutated this tyrosine to an alanine, you get a mutant receptor that mimics the state of elevated cholesterol. And as you can see here on the left, it mimics, like if you have excess cholesterol, everything is shifted to the right, whereas we don't see the same effect when we look at the CCK2 receptor. There's even more evidence that cholesterol levels and CCK somewhat influence each other. So there has been a study with leukocytes that have been derived from patients, and they overexpressed CCK1 receptor and tested for CCK responsiveness. And they found that there was a reduced responsiveness for patients with obesity and diabetes. Another study has shown that there's a CCK1 receptor dysfunction in human gallbladders that have cholesterol stones. So the metabolic phenotype seems to correlate pretty well with CCK1 receptor function or dysfunction. With that, we formed a novel strategy. So our hypothesis is that we need a clean PAM. So that means a positive allosteric modulator without any intrinsic agonist activity. And we think it would have advantages over full agonists at CCK1 receptors. So we might have an angle on an improved safety because when the PAM binds, it would amplify the effect temporarily, and it's correlated to the release of the endogenous CCK after a meal. And it also should not induce desensitization or downregulation of the receptor, which often is also causing issues in the safety and toxicity profile. We could also expect increased efficacy since it enhances endogenous CCK effects, and importantly, it could also correct this abnormal CCK stimulus activity coupling that is particularly found in patients with elevated membrane cholesterol levels. So it could be particularly useful for our patient cohort of interest. So our goal is to discover, validate, and characterize small molecules targeting CCK1 receptor with a novel pharmacological profile, which is a positive allosteric modulator or PAM without intrinsic agonist activity. With that, I want to move into the next part, talking about the screening, the testing funnel, and assay development. So before we start with the screening campaign, we design a testing funnel. In this case, we want to have a testing funnel that will lead us to a clean CCK1 receptor PAM. So as primary screening assay, we're using CCK1 receptor PAM, IP1 immunoassay, looking at the GQ coupling. Then as a counter screen, we want to also look at GQ-coupled GPCRs in endogenously expressed in parental cells. And importantly, we need an agonist assay so we can filter out the compounds that have intrinsic agonist activity. As secondary assays, we can have an orthogonal assay like calcium flux that also is downstream from the GQ signaling event, looking at target engagement with TFA binding. For the GS coupling, we can have a CMP assay, and we can also look at beta-arrestin recruitment. For the scaffold validation, we would do analog by catalog, so looking for commercially available compounds, perform structure-activity relationships, mechanism of action studies, in this case also check if we see activity in high cholesterol environment and at the Y140A mutant. So with that, we hope that when we screen our library of 360,000 compounds, we can identify small molecule scaffolds that could be further developed as a treatment. As I mentioned, our primary assay, we said we would look at, because CCK1 receptor preferably couples to GQ proteins, we wanted to look at the IP1 assay. You can also look further downstream of GQ coupling to calcium, but we decided to use the IP1 assay. When you go the signaling cascade from after GQ coupling, we have the phospholipase C activation, which cleaves the PIP2. We get IP3, which gets degraded to IP1. And this can be detected with the kit from Cisbio, where we have an anti-IP1 peptides on antibody. When it binds to the IP1 that has the D2 fluorophore, we get a FRET signal. And with increasing levels of IP1, we lose that signal. So we have a loss of signal immunoassay, which captures the increased IP1 in cells. As I mentioned, we could also look at calcium flux that is after the IP3 binds to the IP3 receptor, but there have been several reasons why we chose the IP1 assay over the calcium assay. I'm not going too much into detail here. One reason is that it seemed just way more high throughputable in our systems that we had, and also the IP1 assay is an equilibrium assay in contrast to calcium. So we had a specific concept in mind for designing the primary screening SOV. So we were thinking our PAM might silently bind to the receptor and should only enhance the IP1 accumulation in presence of the orthosteric ligand CCK. So this is the screening procedure from this screen that we did in 2019. So we started with the 1536 well plate and had our HEC293 cell line overexpressing CCK1R. We froze a large batch of this monoclonal cell line and pulled out ready-to-use cells as we needed them, plated them overnight. And then we would use our echo that we had to dispense the compounds and let them incubate for 30 minutes at 37 degrees. And then we would move to the Beckman bioreactor that we had at the time and add simultaneously CCK8. So what we do is like using an EC20 concentration, so a sub-threshold concentration of the peptide. So we could see if our PAM that should only act in the presence of our orthosteric ligand shows an effect. At the same time, we are adding the lithium chloride, which inhibits the degradation of IP1. That's why I wrote that here we're pretty much starting the reaction and before we are more silent, because before the IP1 gets degraded, but once we add the lithium chloride, this is when our IP1 signal should be stable over time. So we incubate for another hour, then using the combi, we add the IP1 detection reagent from Cisbio, and after another hour, we moved into our Ferrostar FS using the HTRF module. Since it was a large screen, we had some data processing and analysis in place like GeneData, CBIST, and TIBCO Spotfire. And this assay design was suitable for our high-throughput screening robotic system. In general, we had the 360,000 compound library, so specifically the NIH Molecular Library Small Molecules Repository, or MLSMR. We were able to screen 20 plates per day, so we finished the screen after 14 working days. And as an internal control on every plate, we also included this 6-point dose response of the CCK peptide. And I will come to that in the next slide. So this is an example of our day 14. And we would usually, so we're looking here at 6 plates. And we would plot this dose response curve, so it's a loss of signal assay. This is the raw data from the HTRF module. We have our negative control here, which is the EC20 of CCK. And what was really important is to make sure that we hit an EC10 to EC30, to make sure that because our PAMs should not have intrinsic agonist activity, they can only work when we have a concentration and orthosteric stimulation that is sufficient for them so we could read them. We have the assay window, which was okay. The signal to background was 2.3 to 3. But since the assay was so clean, our C factor was still very good with like a 0.8, around 0.8. This is from the whole screen. We have the distribution histogram. In general, the screen performed very well. We have also a nice normal Gaussian distribution. In blue, we see our compound distribution. In red, we have our negative control. And in green, we have our stimulator control. So one thing that we usually do when we have a screen that involves TF-ret or fluorescence is looking at the F ratio. So here, this is a scatter plot where we have the F ratio on the y-axis and the activity on the x-axis. And we can see that there are quite a few compounds that if we see increasing activity, it actually also see increasing F ratio. So the F ratio is showing the fluorescence interference, and it looks from the compound to the negative control whereas from the TF-ret donor channel. And so we think that might be an indication that these compounds actually have a fluorescence interference. But at this stage, we did not exclude this compound because we didn't want to exclude too many compounds at this stage, but we would look at this again later. So in general, just summarizing the primary screening performance, so we did the 360,000 library at around 6 micromolar, and the C factor was really good, and we got like around 5,000 hits, so 1.6% hit rate. For the hit confirmation in triplicate, we then selected 646 hits. In general, because we had such a robust performance, we felt like we could be pretty lenient with our selection criteria. So the next step would be a counter screen. So we wanted to see which compounds would be assay artifacts or non-specific activators of the IP1 pathway. For a PAM assay counter screen, it's really important to have adequate basal stimulation in the same cellular background. For that, we used a GQ-coupled GPCR that is also endogenously expressed in HEC293. We used the P2Y-R family, like 1, 2, and 11, because they are also GQ-coupled and can be activated by ATP. And this way, we could mimic similar basal IP1 levels to find the non-specific activators. Here on the left, we see the control curves, also great performance, and we get like a nice in negative control basal stimulation around the EC20, EC30. And furthermore, we are around the same range of our TF-ret ratios, which can translate to the IP1 levels. So this performed very well, and we were able to filter out quite a few compounds. So at the end, we ended up with 296 hits. And I'm coming back to the F ratio here. So on the left, we have our CCK1 receptor PAM activity. On the x-axis, on the right, it's our counter screen activity. And I think you can clearly see that we get this similar pattern here with the F ratio for quite a few compounds. So there's an overlap of the counter screen active hits and compounds with high F ratios. So our counter screen actually detected the assay artifacts and the non-specific activators. So the 296 hits that we selected were in this little corner here. And for hit prioritization, we would perform 6-point dose responses from liquid stocks. So for the hit prioritization, so we did perform the P2YR counter screen, we did the CCK1 receptor PAM primary assay, but also importantly, we wanted to do the agonist counter screen to make sure we're not getting compounds with intrinsic agonist activity. We performed it in the same assay, but in this time we did not have the EC20 concentration of CCK as an orthosteric stimulator probe. And then in the scatter plot, we compared the maximum efficacy from the PAM on the x-axis, and on the y-axis we have the agonist maximum efficacy. And you can see that almost all the compounds show great correlation. So the majority of hits show intrinsic agonist activity. And I had labeled some of them because I will come back to them later, hits 8 to 15. And then we have a few hits around here, the hits 1 to 7, with no or minimal activity in the agonist format. So these have the potential to actually be clean PAMs. And we've developed a binding assay in hope that it could further support our hit prioritization. So I want to go a little bit in the assay development of our TFR binding assay. And the approach, it uses a SNAP-tagged receptor. So we need a HEC293 cell clone expressing the SNAP CCK1 receptor. And the SNAP is on the end term of the receptor. Then we would label it with lumeforterbium cryptate. And at this stage, so we have now our TFR donor on the receptor. At this stage, we perform a membrane prep, and we can freeze it and pull it out whenever we need. So for the assay, we pretty much only have to add our hot and cold ligands. So the hot in this case is a fluorophore labeled CCK derivative. And when it binds, we have the proximity of the donor and the acceptor fluorophore. And in this case, because we used a green fluorophore, we used the Lantus screen module from the Ferrostar reader. And to finish our assay setup, we needed to design our fluorophore labeled peptide and also optimize the assay conditions. So from the two parts for our binding assay, one is the SNAP-tagged receptor, and we always wanted to make sure that the SNAP tag is not interfering with the functionality of the receptor. And in blue, we have our SNAP CCK1 receptor cell line, and in black, we have the wild-type cell line. And the SNAP CCK1 receptor is still functional, and the potencies are within range to the wild-type. So we felt confident about introducing the SNAP tag to this receptor. For designing the fluorophore-labeled CCK, I like to look at, if available, the cryo-EM or crystal structures. And it's easier, obviously, if you have a peptide GPCR, because usually for these peptides, there are multiple ways to introduce a fluorophore. In this case, we saw that there's a free amino group of the CCK on the end term, which is suitable to introduce a tag. We worked with ANASPEC, which helped us to ligate the highlight fluorophore 88 to a version of CCK8 that has some modifications, like the threonine and nucleosine on the CCK peptide. And it's a non-oxidizable version, so it is more stable, and we thought it's probably a good way for to put it in our automated binding assay. So having both parts in place, we then could perform saturation binding experiments. So we have here the non-specific binding and the total binding. It's a beautiful saturation binding curve. Somehow it liked the two-side model better, so we have a high and low affinity state. But it worked beautifully, and we could then also use the KD that we get from our fluorophore-labeled CCK to perform competition binding experiments. And so for CCK1 receptor, there are multiple ligands available that we can test. And we have CCK8 and CCK9 as full agonists. JMV180 is a partial agonist, and diversified is an antagonist. And looking at the binding affinities we got with our TFR binding assay, compared to the binding affinities from the radioligand binding assays, they're very similar results. So it seems to work very well. And even though I'm not going to show it in this presentation, but this TFR binding assay is really also suitable for high throughput kinetic experiments where you can read in one well the fluorescence over time for association binding and dissociation binding experiments. So we optimized this assay for automation. It can be run in a 1536 well format, and it has a very straightforward SOP. So with that, we tested the 296 hits in triplicates that we had for our hit prioritization. And what we found was pretty interesting. So the subset of hits 1 to 7 that had no to minimal activity in agonist mode, none of them showed any effect on the binding. In contrast to that, the compounds or hits that were active in agonist mode, the majority of them showed reduced fraction of the hot ligand bound. So very likely we have orthosteric displacement here, which also goes well with the idea that we have here agonists. But in theory, it also could be a negative allosteric modulator for binding. So the CCK1 receptor is really interesting in the sense that it has like this very distinct low and high affinity binding states. So because some might wonder, should we not see an increased binding with our PAMs? And we are actually not that sure about it, because what they found with cholesterol at CCK1 receptor, that actually enhanced the binding. So we actually see a shift to the left, while it reduced the GQ-mediated downstream effects. So we were actually not sure if we even wanted to see an increased binding with our PAMs, because that's more correlating to negative effects on the downstream events. So I think what this binding assay did is really kind of supporting our theory that we have orthosteric agonists that were like the majority of hits that we had found in our screen. So this is summarizing the first part of the screening and hit prioritization. So we did the 360,000 compounds at the primary screen, hit confirmation, and counter screen ended up with 296 hits. And then moved into hit prioritization with the 6-point dose responses for the PAM and the agonist and the counter screen, and then did the competition binding. And because of mainly so many of these compounds had intrinsic agonist activity, we only had 45 hits to move forward. 36 of them were commercially available, and we wanted to go into the hit validation mode here. So before I go to hit validation, I want to go in another secondary assay that we had designed for hit validation. As I mentioned, CCK1 receptor couples to GS protein and recruits beta-arrestin at higher CCK8 concentrations. We usually use a CAMP immunoassay that works pretty similar to the IP1 assay, like GS stimulates adenylate cyclase, increases the CMP levels, and having the D2CMP and the anti-CMP cryptase having a TF-red loss of signal assay here. For beta-arrestin recruitment, we have started to use the nanobit system. So we have the small bit tag, the 11 amino acid small tag on the C term, whereas the large bit is fused to the beta-arrestin. If we have an agonist that binds to the receptor, the beta-arrestin gets recruited, the split luciferase finds each other, it complements, we have a function luciferase, and when we add substrate, we can read luminescence. In theory, there are 8 combinations of the small bit and large bit placements possible, but we found that the small bit on the C term of the receptor and the large bit on the N term shows the best performance. During the development of these 2 assays, it appeared to us that the assay conditions on the timing actually appear compatible to do some sort of multiplexing. So for that, we had to get a few more details in place. So first we wanted to know, okay, for the beta-arrestin assay, because we can record a kinetic time course, we wanted to know where is our peak. and we have the luminescence on the y-axis and the time on the x-axis here, and we found that the luminescence peaked after 5 minutes. The other thing we wanted to ensure is that our CMP response in the nanobit cell line, so in the cell line with the C-terminal tag on the receptor and the beta-arrestin large bit, actually shows similar effects than our wild type. And we do see a slight rightward shift in the nanobit cell line, but we thought it was still in the acceptable range to move forward with our experiment. So the procedure that we came up with to read this, both the CMP and the beta-arrestin in the same well, is as follows. So first, we have our nanobit cell line and the substrate in buffer, and we let it equilibrate for 2 hours at room temperature. We have our echo, and we dispense our ligands and compounds, and IBMX. So IBMX is a PDE inhibitor, working similar to the lithium chloride, but for CMP, stopping its degradation. And everything was dry dispensed on the 1536 well plate. We then moved to our combi and dispensed the cell line with the substrate. And at this point, when it then meets the compound and ligand IBMX, this is when we start our CMP and beta-arrestin assay time. And after 5 minutes, we would go to our Ferrostar and read luminescence to get our beta-arrestin readout. And after 30 minutes total, we go to the combi, dispense our detection reagent from the CMP kit, and after 30 more minutes, go back to our Ferrostar and use the HTRF module to get our readout there. So the curves that you can see here are recorded from the same plate, from the same wells, using the same ligand droplets, same buffer conditions. Everything is the same except the readout. So we have the CMP in the squares, and in the circles we have beta-arrestin. And interestingly, as I said, GS and beta-arrestin is actually not the predominant coupling usually for CCK1R, but interestingly that they really had pretty similar potencies. So we saw that even the EC20, which we use for our PAM formats, were very comparable. So we actually could use this multiplex format to also screen for our PAMs. With that, I'm gonna quickly move to the hit validation of the original hits. So we obtained 36 compounds to be tested as dry powders, but I wanna focus on the 4 most promising analogs. So we have here hit 1 and 6, which have a tetracyclic scaffold. And interestingly, also hit 3 shows this tertiary basic amine, so we think that actually could be an indication that this is important for our allosterotic binding site. Here we have the IP1, CCK1 receptor IP1 PAM assay in 16-point dose responses. In the middle, we have our agonist, and on the right side, we have our parental cells. And we see substantial activity in our PAM assay. It's dead in the agonist assay, and except for hit 7, they are all dead in our parental cells. We also tested in our multiplex format, so for CMP, we see that 3 of the 4 compounds show also a nice activity here. For the beta-arrestin, most of them don't really show activity except hit 6 that shows slight activation. And as also shown with the single points, we don't really see any effect on the binding. So overall, this data suggests that we have a clean PAM that mainly that can positively modulate GQ and GS protein signaling. So to finalize our testing funnel, we have now the hit validation. We have the 6 assays that we performed, all performed with great C factors. Everything was performed in 1536 well format. And at the end, we had 4 hits and 3 scaffolds. So now I'm going to the last part of the talk, which was published in Membranes recently. talking about the mechanism of action studies that we did with our tetracyclic analogs and some structure-activity relationships that we found. So for that, I want to go and we have go a little bit more detail. So what pharmacologic profile are we actually looking for? So, and we found 4 main criteria for the scaffold validation. Obviously, first, we need substantial positive modulation of the CCK-mediated CCK1 receptor GQ signaling. Second, we don't want to have agonist activity and also no receptor internalization. The third criteria is that we want to maintain our PAM profile in an environment with elevated cholesterol. And the fourth is that we want the compounds to be selective to CCK1. And I already mentioned we have the parental cell line that expresses endogenously the P2YRs. We want to check that we don't have cross-reactivity with CCK2 receptor. So these are the IP1 and calcium flux assay readouts for GQ signaling. But since we also found effects on, for the GS coupling, we wanted to also use a receptor that predominantly couples to GS, like the vasopressin 2 receptor. And this was our selectivity panel at this stage of the project. So our collaborators tested the compounds on their CHO cell line that expressed CCK1 receptor or CCK2 receptor. And what they did, we have here a dose response of CCK peptide, and then they add a single concentration of the hits, hit 1 and hit 6. And we found that there was a 3 to 8-fold leftward shift in the calcium flux assay. So also confirming that we have PAM activity in calcium flux and not only in our IP1 assays. We don't see any effect in our CCK2 receptor cell line. There's minimal agonist activity at very high concentrations, and it's stayed in the CHO parental cell line. So what we wanted to see, is there potential for an enhanced safety and efficacy profile? So one thing we were interested in was CCK1 receptor internalization. So the top row, we have the CCK peptide, and you can see that the receptor is at the beginning, it is on the membrane. When you add CCK after 15 minutes, it's pretty much all removed from the membrane. Whereas if you use 10 micromolar of hit 1 or hit 6, we don't see any receptor internalization. We were also interested to see how are these PAMs performing on our Y140A mutant receptor or in an environment with elevated cholesterol. And we were excited to see that our PAMs maintain the PAM activity at the mutant and also in an environment with elevated cholesterol. And it could even correct the CCK8 dose response, so leftward shifted, so that it gets to potencies as cholesterol-free levels. So one thing for the allosteric activities, we have certain parameters. So one thing is to look at probe dependency, and we wanted to see, are they just working in cooperative with CCK or also some other CCK receptor agonists? And here we have a partial agonist, and we see that there's a 5 to 6-fold shift, but also a 20% increase when we add our PAMs to the compound. It's not shown here, but also in the paper we described that when we add our PAMs to CCK33, we're actually seeing a 12-fold leftward shift of the curve. So there's some probe dependency going on, but it seems like they seem to work and positively modulate across the agonist panel. One thing is also using the operational model of allosterism. So we have the dose response of CCK, and then we add increase in concentrations of hit 1 or hit 6, and then we can use our equation to calculate either the KB, what is the affinity of our PAM with the allosteric binding pocket, and cooperativity factors alpha and beta. And we have the affinity of like 25 to 31, so it's low, but it's significant affinity to CCK1 receptor. And more importantly, we have substantial positive cooperativity. So everything above 1 is considered to be a PAM, and we have 10 and 31, so we definitely have positive cooperativity here. We have tested HIT1 and HIT6 for selectivity against the P2Y receptors and CCK2 receptor. So when we moved and actually tested those compounds in an AVP2R expressing assay, we unfortunately saw that especially HIT6, so we have in black CCK1 receptor IP1 PAM activity, and in AVP and in red we have the vasopressin 2 receptor CMP activity, and our HIT6 has really a strong off-target effect on that receptor. So for that, when we then used and did the analog by catalog and our structure activity relationships of 65 compounds, we definitely moved in our vasopressin receptor assay to monitor and see if we see this off-target effects quickly and if we potentially could eliminate them. We also checked obviously for the PAM activity and wanted to make sure there's no intrinsic agonist activity going on. I'm just quickly going to summarize our SAR. So we did multiple modifications and found that compound 2 actually is an improved version of our hits. So we had checked different amines, and we found that it's pretty crucial to have that. And also this moiety here seemed to have the best profile. As well, the morpholine here showed generally the better pharmacologic profile. But the most important part was really to see that this dimethylthiocyclohexyl, so here in orange, was able to completely eliminate the vasopressin 2 receptor off-target effects. So we have here again HIT1 and HIT6. And now looking at the 2 compounds that we screened that had this moiety, and we can see that the AVP2R off-target effect is completely eliminated, and these compounds are now selective to CCK1 receptor. So with that, I want to conclude. We screened our screen library, had our testing funnel. We had designed a comprehensive toolbox for each of these stages. We successfully screened. We think we got a strong testing funnel because we were able to identify the tetracyclic scaffold with the desired pharmacological profile. We had looked at the 4 criteria for the scaffold validation, and we were actually able to obtain CCK1 receptor selectivity, along with being active in elevated cholesterol levels, having no or minimal intrinsic agonist activity. And the only thing we think we should maybe improve further is actually maybe getting better potency in our CCK1 receptor PAM assay. But we think that a targeted MedChem campaign could improve this and further develop this scaffold as a potential therapeutic for obesity. With that, so I want to thank my ex-colleagues from Sanford Burnham Prebys that have worked with me on this project, our collaborator from the Mayo Clinic, the funding from NIDDK, and I want to thank you for your attention.